Required practical summaries. There are three that are common to both triple and to combined. And these are the three. The first one is the reaction time required practical. It's a really straightforward practical, but because it's a straightforward practical, it doesn't mean it's easy. There are lots of questions they could ask about it, and they could link these questions into in other questions around the nervous system, which makes it a little bit tricky. So we'll have a look at some of those questions. The other practical is 2-in-1, which is field investigations, but that's made up of random sampling using quadrats and also random sampling using transect lines. And again, people are quite good at random sampling using quadrats. Lots of time is spent on that in class. Generally, in my experience, transacts get less of attention in class. So it will be an idea to just make sure we're familiar with transacts and how those work. Pretty straightforward, and then we'll look at some common questions for those. So that's what we're going to do. As I say, it should take less than the hour to give us more time. What we'll do, once we've done the practicals, we'll have a short break, for just a comfort break of 5 minutes, seeing how we're doing, and then we'll come back in to do the second hour. Okay, so first one is reaction time. So in the reaction time required practical, what you have to do is you have to carry out an investigation. So you have to be able to plan this. So they're very specific in the specification. You must be able to plan an investigation. So you will need to write a method if this comes up. Into the effect of a factor, okay? Now it is clear on the specification that it is a factor. Now this is the practical that we do in school, that's advised by AQA, but the fact that the spec says a factor means that actually they could ask you about changing other things. So I'll get into that in a little bit more detail. So I've done the basic one that you'll have probably done in school. The thing I am changing, the independent variable, is just going to be the person catching the ruler. So we're just going to start out with two people. We've got person 1 and person 2. All right, very exciting names, but that's what we'll refer to them as. The key thing is both of them have got to use their dominant hand for this practical. So that's going to be a control variable, making sure that they are using their dominant hand, so that they've both got a similar level of competence with their hand. If you have one person using their dominant hand, one person using their non-dominant, you're going to be changing two variables. So we're going to have the dominant hand. Now, this bit's a bit weird and it sounds a little bit overly instructive, but there's a reason for it. The next person is, you start with person 1, and it's told they're to sit with good posture and have their forearm on the table with their hand overhanging the edge. And that's basically, the reason for that is because it locks the hand in position. So often when you see this in school, you see, I've observed quite a few lessons where the teacher's done this and they've not followed the method brilliantly, and they've just got all the pupils to sort of stand up and drop the ruler and the rest have to catch it. And what you see is people running over going, look, my reaction time's 0. And it's because they watch the ruler and they move their hand down and catch it lower than they would have. All right, so that's going to give you an accurate result. So they're very clear in the specification. You have to specifically say, put your arm on the desk with your hand overhanging. If you got asked a 6 marker on this one, in order to get a valid result, you would have to mention how you're going to lock the arm in place, because otherwise you're going to have inaccurate results. So you get capped at 4 marks. Okay, the next thing is that the person holds the ruler vertically. Again, that's a control variable. If you put it at an angle, it's not going to affect the results. With the 0 centimeter mark between the person's thumb and forefinger. So as a control variable, you're just having the same starting position for each person. That's the whole point of that. And then they tell person 1 to prepare. What they don't do is they don't give them a countdown. They don't say, I'm going to drop the ruler in 3, 2, 1, are you ready? Because then it's not reaction time. The person knows it's coming. They just say, I'm going to drop the ruler at some point. Are you ready? So that the person isn't looking around the room or distracted by what else is going on with another group. And then obviously that's the issue. You've got a distracted person. So it makes sure that the person is ready. But then they do have to release it randomly. Okay, they drop the ruler, the person will catch it as quickly as possible. And then what you do is you record the number on the ruler at the top of person one's thumb. So I've drawn a diagram here to show that, because that is a common error that might get shown up in an exam. If you recorded at the bottom, then that's not actually a reaction time. That's going to show you too low. So we must record at the very top of person one's thumb, which is why we tell them to catch between thumb and forefinger. If they catch with their full hand, then it's a bit harder to tell where the reaction time actually was. So we stick it into a table like this. So you've got person one, so their first attempt, they got 27 centimetres on the ruler. What you then tell them to do is to have a little bit of a rest, and you're just going to repeat it several times. Now I've drawn it 3 times, that's because the only space I had available. But for this one, it's quick. You want to be doing it 5 to 10 times. So you'd say do it at least 5 times. And then obviously you're going to calculate a mean, because that's what we do in all science experiments, isn't it? We calculate means. We obviously look for inaccuracies. So this might be an opportunity then for them to give you a set of data. Maybe one of them's anomalous. It'll be really obvious if it is. Don't try and make things anomalous just because you've heard about it. It'll be really obvious if it is anomalous. And then you calculate the mean. So that is like this bit here, 1 to 6, is like the standard method for testing anyone's reaction time. What we're now going to introduce is the effect of a factor. Now, for this method, I've just got the person catching the ruler. So basically what happens is person 2 swaps over. Then now you've got a different person. So that was your independent variable. It was the person catching the ruler. And you just do the whole thing again with person 1 dropping it. And you get person 2's reaction times. But at this point here, step 7, we could have changed that for anything. So we could say switch roles, and then a person of a different gender catches the ruler. That way you're investigating gender. Or we could say switch roles, and the second person is a lot older than the first person, and then that way we're investigating age, on effect on reaction time. You could say, right, I want to keep the same person. So you've done this steps 1 to 6, and they've always been catching with their dominant hand. Let's say they're right-handed. And then you would say, right, for the next set, we're going to do it again, but this time you're going to use your left hand. You're going to use your non-dominant hand. So in that case, we're investigating dominant versus non-dominant hand. They could also do, and this is the absolute favourite that they love to ask about, is caffeinated or not. And it very rarely gets done in a lab. When I first started teaching, I used to do this experiment, but then so many people were allergic to caffeine, it just became a thing that people just don't really do in school. So we used to do this experiment with the reaction time exactly the same way, but when we get to step 7, rather than just switching people, we do something a bit more exciting. We give the first person, the person who's done the catching, we give them a drink of Coke, all right? Or we give them a drink of non-caffeinated Coke sometimes. And then what you can see is you can investigate whether or not caffeine affects reaction time. And we're actually going to have a look at that question. But the exams love to ask about caffeine and its effect on reaction time. So once you've got all of the person's ruler measurements, what you do is you take the mean, and then what you have to do is you have to use a conversion table to look up their reaction time. So you can see in this space here, person 1 got 27, 23 and 19. So if we just very quickly did that mean, so 27 plus 23 plus 19. Please make sure you show you're working out, because I have just done some more training with the exam board, and they were very, very specific this year in saying that if you just have an answer without any working out whatsoever, that will get escalated, and the head examiner will look at your paper, all of your paper, because they want to know whether or not you've written that and if you've been cheating. So please show you're working out. You must show where your answer comes from. So 27 plus 23 plus 19 divided by 3, and I did this earlier, so I'm not a maths genius, I just know the answer, it's 23. So then they would go to the table and they would look that up and say, oh, okay, this person's reaction time is 0.22. So that is the reaction time required practical. It's quite straightforward, but as I say, they could ask about any type of factor that would affect reaction time, and you must be able to plan a method for it. Key thing, is just what you're changing in step 7. I will share this workbook with people. There's also a link on my Facebook science course you paste to a YouTube video where I pretty much do what I'm doing now. I've explained the whole thing, so I'll also drop that link in, because that will have this video and also the explanation. You can watch it back if you want to. Right, here are some common questions. So this first common question is just giving you a set of data. I'll try again. Giving you a set of data. So you are given the set of data in a table. This is just basically saying, can people in this exam understand what tables are showing them? And hopefully you can. So this is about reaction time. We've got a before, we've got an after, and then we have got 1, 2, 3, 4, 5 different students who have done this. Okay. So this makes our results a little bit more reliable, because we've got 5 different people. And all it asks us is, it says, what is the effect of drinking coffee on reaction time? So again, they're asking about caffeine. They love to ask about caffeine. It's because examiners are fueled by it. So we can see that before drinking coffee, person 1 had a reaction time of 385. After was 255. So we can see that the reaction time has decreased. 420, 291, their reaction time has decreased. And in fact, if we go through them all, we can see that all of their reaction times have decreased. Look at person 4, caffeine monster, massive decrease. So we can just say a simple statement that the effect of drinking coffee is that it reduces reaction time. You might think that is a very simple statement for a table, but if you just look, it's only worth 1 mark. So anything that's just worth 1 mark, you just need a simple statement. All right, so off another paper, so as I say, this is really common what they do. Another data set given to you about reaction time. This time, a little bit more complicated looking table. That's because they've included 3 different tests. I use a better highlighter colour. 3 different tests in there, and then they've got the mean at the end. Okay, so actually, what we need to do is we can just look at this data here. Now, it does not tell us what the factor was here. It just says, here's the reaction time of 2 different people. And it doesn't say what factor is affecting them. Okay, so we're just being asked to suggest 2 reasons why student A's reaction time was longer than student B's. Okay, so if you look at the data, you will see student A on both methods has got a longer reaction time than student B. So all you need to do is make suggestions why. Now again, really key, it says give 2 reasons. It's worth 2 marks. Really common in the exam is for people just to write down all the reasons that they can think of. And that creates a problem, because the examiner is looking for 2 reasons. If you give 3 reasons and they're all correct, that's fine. All right, you're not going to get any more marks, you'll still just get 2 marks. If you give 3 reasons and 2 of them are correct, but 1 of them is incorrect, your incorrect answer takes away 1 of the correct marks, so you'll only get 1. If you give 4 reasons, 2 of them are correct, 2 of them are incorrect, you're going to get no marks. So do not write something down if you are absolutely not sure. Okay, this would be a possibility. If you wrote 3, cross the 1 out that you are, if you are worried about it. Okay, so don't write more than 2. It says give 2 reasons. So the 2 reasons that affect reaction time, there's lots of them. You could talk about age, you could talk about caffeinated or not, you could talk about practice. The most common ones is you could say that person B, so that's the fast reaction times, person B has taken caffeine. That is absolutely going to get you the marks. The other thing you could say, so we're looking for reasons why person A might be slower or person B is faster, is that person B has had more practice at the method. So this is really common. If you've got someone who, sat there practicing that reaction time practical over and over and over again before they did it, and then they did it, and then the other person has never practiced it, that they're going do better at it. So other things you could have said that person B used their dominant hand, whereas person A used their non-dominant hand. They're all valid reasons, but just stick to 2. Right. This one here is just a link. So when they do this, this is part of the specification. It's part of the nervous system topic, which means that they can ask questions related to the nervous system within the question. So hopefully when you're seeing this diagram, your brain is shouting at you, oh, that's a reflex arc, or it's a reflex response, because that's what it is. And maybe you're even looking at it and naming the names of these neurons, because you need to be able to. It is so common for these questions on reflex actions to come up. So it says, human reactions are responses to external change. Reflex actions help protect the body against damage. So a reflex action is one that is automatic. It does not involve the brain. It is fast in order to make sure that we don't have long-term injury or we don't have death, basically. So what it says is a stimulus from the hot pan. So we can just write stimulus under here. Okay, it's a good idea to sort of scribble on your exam paper. It's not going to lose your marks in order to label a few things to help it make sense to you before you start answering the question. So a stimulus from the hot pan will cause the muscle in the arm to contract and move away. Describe how the stimulus from the hot pan reaches the muscle in the arm. All right, so basically we need to describe all of the stages between the stimulus and the muscle in the arm. A muscle is an effector. Okay, so we've got to say everything in between. So it's just worth, when you're doing something like this, because it is worth 4 marks, that's a good amount of marks, 4 marks. So it is worth labeling this diagram and then ticking off when we've talked about it. So remember, we've got the stimulus. The bit people often miss out is the thumb is the receptor. You have lots of different receptors which detect stimuli. So you could have the eyes are light receptors. The nose is sensitive to chemicals. The skin is sensitive to pain, touch and heat, temperature. So you've got lots of different receptors. Obviously the ears are sensitive to sound waves. So you've got your receptor in the skin. The receptors are then going to pass the information along this neuron, because the information has to travel. Now in the body, we're going to talk about the nervous system in the second session, but basically it transfers electrical impulses. So this neuron is the sensory neuron. The way to remember that is if you think about your senses. So this is a person touching the pan. Touch is a sense. So it's the sensory neuron that's going to pass that information on. This area here is known as the CNS, or the central nervous system. So in a reflex action, the sensory neuron takes the information to the CNS. The CNS decides, is this something that I need to do an automatic response, like this one, or can I send it to the brain? This is needed for an automatic response. So it's going to involve this neuron here, which is called the relay neuron. The relay neuron, its job is to literally pass the information. It needs to pass the information now to something that will cause a response. All right, and that's going to go down here, and it's going to go to this muscle. The name of this neuron is the motor neuron. Okay, the way to remember that is think about cars. What makes the car move? It's the motor. Well, electric cars. Think about something that's got made of electricity, like a drill or electric car. The thing that makes it move is the motor neuron. The motor neuron passes it to an effector. The effector could be a gland or a muscle. In this case, you are very specifically told that it's a muscle, so we can ignore that, but just for reference. And then the muscle will contract and it will move the arm, creating the response. So that would then go to the response. So that is the reflex arc. So now we've labelled it, all we need to do is just add in the description of how the stimulus gets from the pan to the arm and all the stages in between. So because this is a long answer, 4 marks, it's going to require you to write in a logical way, and obviously it's asking for how it gets to one place to another, so it goes in order. So it would be a good time to pull out our bullet points. So bullet point 1. So the first thing is that the receptor cell in the receptor in the thumb detects the stimulus, okay, of the hot pan. We can tick off the receptor, we've done that one. Next, the sensory neuron. So the sensory neuron carries, you can say the information, you can say nerve impulse, okay? So I'm going to use nerve impulse. So the sensory neuron carries the impulse, or you could say electrical impulse, to the, now, CNS. That's just an abbreviation because we can't bother to write it, but it stands for central nervous system. If this is your exam, please write it out properly, all right? So it carries it to the central nervous system. And then what you can do is you can write CNS next to that, because now the examiner knows that you're referring to that. Okay, so another part that's really key with examiners is that if you use the word it, or it's not clear what you're referring to in your answer, they have to sort of give you the benefit of the doubt, but if it's not clear, then they don't give you the marks. So it's worth really stating the names of things, their proper names. So we've talked about the sensory neuron. We've talked about the central nervous system. You would then say the relay neuron transmits the impulse to the motor neuron. Okay, so we've talked about the relay neuron. And then fourth, which will be our 4 marks, the motor neuron carries the impulse to the effector, which in this question you must say is the arm muscle. Okay, that would be our 4th point. We could go on and add a 5th point, which said the arm muscle contracts and moves the pan, but we don't need to, because we just need to say how it gets from the stimulus to the arm. So that is our 4 marks. Really common. Yes, Chris, I'll put the editing notes in there as well. As I said, there's also I'll put a recording of the session in as well if you want to skim back too. Right, you may have to write a method to investigate the effect of another variable on reaction time. Describe a method to investigate the effect of caffeine on reaction time, and it's a 6 marker. So guess what? It's going to be a long answer question. Now, and these are what scare people. You are being asked specifically about caffeine. So you've got to use the general method, which is the one I showed you on the first page, but we've got to change it so that we're not investigating 2 people, we're investigating caffeine versus non-caffeine. Okay? And it's a description, so again, we don't need to explain it, we just need to give a step-by-step points. Now, before I start and just show you the method, or talk you through it, I'm not going to write out a full method. You're not on here to watch me write a method, I'll just give you the method. But what I want to talk about is about how they mark 6-mark questions. So 6-mark questions, the first thing the examiner will do is they will read your response and they will put it into category 1, 2 or 3. Category 1 means that your response can get 1 to 2 marks, category 2 means it can get 3 to 4 marks, and category 5 means it can get 5 to 6 marks. And they'll have qualifiers on those, okay? Now the key qualifier is this. For 5 to 6 marks, it needs to be a logical method, and it needs to lead to a valid outcome, and you've mentioned all of the steps. So basically, if your method would give 2 sets of data, which would show you the difference between someone who has had caffeine and someone who hasn't had caffeine, and you could work out reaction time from it, they have to put you in the 5 to 6 mark category. 3 to 4 would be not necessarily a valid outcome. Now, what that means is that you might actually get a valid outcome, but you can still be put into 3 to 4 if you haven't got all of the steps involved. Right, so you might not necessarily, so you don't need to actually write the proper practical out to get 3 to 4 marks. all right? But you do need to identify many of the steps, and it does need to be logical. So basically what happens here is people normally get in 3 to 4 marks because they've just mentioned lots of the right things, but actually they've got a bit muddled up in between when they're writing their answer. And that's because people normally just they just start writing and write a massive paragraph. I'm going to suggest that you don't do that. Please use a bullet point list, and you're more likely to jump yourself into this category. Question for 1 or 2. All right, first off, so you've put something, so you've just written something valid. All right, you're not necessarily got the right outcome, but you've written something like they should drink coffee and 1 shouldn't drink coffee. Oh, what, you've got a mark. Okay, so you're going to get 1 or 2 marks, but you've not got the right. You wouldn't actually find out anyone's reaction time if they followed your method. You've just mentioned some things that are correct, like measure it using a ruler. That's it. So here is the method. Now, you can figure this out just by going back to this method here. I'm just going to do this method, but then when I get to step 7, I'm just going to repeat. I'm just going to change it for caffeine. That's the method, all right? So I've written it prior, so I'm just going to do this and move it. Oh, no, I'm not. I'm going to try and select that box somehow. There we go, and I've got rid of it. It's not what I want to do. I'm going to try and select that box and move it. There we go. So first off, we get person 1, and they sit with good posture, forearm on the table, dominant hand overhanging the edge. So we have given all the qualifiers we need for a nice controlled test. So, because we want the 6 marks, don't we? We want a logical, valid outcome. So we have to mention control variables. Person 2 holds the ruler vertically with the 0 centimetre mark between person's 1 thumb and finger, and instructs person 1 to prepare. All right, again, exactly the same as the basic method. Person 1 catches the ruler as quickly as possible when it drops. And we record the number at the top of person 1's thumb. And here is a picture just in case you don't know what a thumb looks like, so you can see the top of it. 5, you let them rest and you repeat it 5 times and calculate a mean. You could write 10 times, just a sensible amount. 6, convert these ruler measurements into reaction times using a conversion table. So that is the perfect method for finding out one person's reaction time. But we have not changed anything at this point. So all we need to do now is change something. So person 1, so it's the same person, because we can't have one person who is caffeinated and one person who isn't, because then we're changing the person and the caffeinated, which we can't do. We only change one thing. So it has to be the same person. So the same person 1, now drinks a caffeinated drink. Give them 30 minutes to let the caffeine get in their system. And we repeat steps. Now I've made a mistake here. I wrote steps 1 to 7, but actually it just repeats steps 1 to 6. So just say repeat steps 1 to 6 to find out their new reaction time. You don't need to do anything more than that, okay, because you don't need to go in and start writing the method out again. So that is the method of how you would find someone with caffeine. That would get you 6 marks. The reason why bullet pointed lists are so good is examiners, we love them because we can tell very quickly what category we're going to put you in. And when we see them, we pretty much go, this is a good one. All right, you put yourself in their mind going, oh, look, he's used a bullet point list. This person knows what they're talking about, even if they don't. So it's just better when you see them. But if you make a mistake like I did here, it's very easy to figure it out. So repeat steps 1 to 7. Obviously, I can just cross that out and just write 1 to 6. It's not lost in the middle of a massive paragraph, okay? If I'd forgot something from here, so let's say that I'd forgot to put in this step here, I forgot to put any repeats, and I could just add it down here. So normally when you write a big load of text, you have to put an asterisk in and then add that in somewhere else, and sometimes when they scan your papers, that could get lost if you've got it on the additional sheet. If you put a bullet list in, it's very easy. You just can change the order of your numbers to make sure that it's logical. So really good. Please use a bullet point list when writing methods. Okay, right, field investigation. So there's 2 practicals here, nice and quick. The first one is random sampling. So you need to estimate population sizes of plant species in a survey area. All right, it specifically says plant species on the spec, but I have seen questions about animal species. All right. So the first thing we need to do is we need to measure the length, width, and area Now, this is where you need to mention equipment, because remember, it needs to lead to a valid outcome. So if you don't tell them how to measure it, you can go wrong. If you say a ruler for measuring the length of a field, you will not get the marks, because that is not a sensible piece of equipment. You're very likely to make mistakes if you go around measuring the size of your school field with a 30 centimetre ruler. So you'd use a tape measure or one of those trundly wheel things. So, measure the length, width and area being sampled with a tape measure. Calculate the area in metres squared using length times width. That is obviously if you have got a square field, which most of the time they do give you. Notice there's a question over here where it was on a higher paper where the field is not square. All right, but it's still pretty easy to work out the area because we were given a nice square or a rectangle here, and we're given a nice triangle here. So you just have to use some maths in order to figure that out. Okay. Once you've figured out the area of the field, what you then need to do is you need to figure out the area of your quadrat. Now, this is where they put in some complicated stuff, because the area of the quadrat that they normally give you is 0.5 by 0.5. And lots of people look at that and go, oh, 0.5 and 0.5, so the area is 1 metre squared. No, it is not. If you do 0.5 times 0.5 on your calculator, you will get the answer 0.25 metres squared. Okay, so my advice there is always use a calculator, even for the things that you think are really simple. That's going to make a massive difference to our final answer. In fact, if you make that mistake, you will definitely lose marks. Place the quadrat randomly in the sample area and count the population of species, e.g. daisies, right? It is so important that you use the word randomly. Now, in your method, you do not need to explain how you are going to place it randomly. It wouldn't be a bad idea to, but you don't need to get into too much detail, okay? But you might get asked about it, so I'm going to get into that in a little bit. So place the quadrat randomly in the sample area and count the population of plant species, e.g. daisies. So let's say we had a big field like this. Okay, the way to get randomness is to divide your field up into sections the size of the quadrat. Okay, so let's say that we did this and we had 100 sections, and you just number them 1, 2, 3, obviously 4, 5, 6 to 100. And then you get a random number generator and you just choose 5 random numbers. Or it said 10 here, so 10 random numbers. And that's where you put your quadrat. So that's how you get randomness. That's the easiest way to do it. So all you need to do is you don't need to explain that. So you just say put the quadrat randomly using a random number generator, and then count the number of plants that you find in there. Okay, you do it 10 different areas of the field, and then you calculate a mean. So again, this is one where they're going to get you to calculate a mean. So let's say that we did this, and let's say that our mean was we had 8 daisies. It could be buttercups, it could be anything. All right, that was our mean. Then what we do is we use this to find out the number of daisies, or estimate the number of daisies in the full field. All right, so very straightforward. Estimated population size is the total area of the field. So whatever the field was, so I'll give you an example. Let's make it nice and easy. Let's say we had a field that was 10 meters by 5 meters. So our total area of our field is 10 times 5, so it's 50 meters. So it would be 50 divided by the area of the quadrat. We've already worked that out, which was 0.25. So that's going to tell you how many times your quadrat will go into your field. So in this case, it's 200 times. So you'll have 200 quadrats will go into this field. In each of those quadrats, there is 8 daisies. So all we do is we times 200 times 8, and that will give us our answer. So that should be, if my maths is correct, 16,000 daisies in the full field. Please do not do what I've just done and do mental maths in your head. You've got a calculator, use it, even if you are convinced you know the answer. All right, because we don't want to make a maths error and lose some marks when your biology is good. So that is the method. What kind of things might they do? So I've already given you this one. They might ask you to find the area of a field that isn't a rectangle or a square. So you'll have to think about it. all right? It might be any random assortment of shapes. It could even be this shape, all right? And you could just divide it into triangles and squares like you would do in maths to figure that area out. This section here is just talking about how to place it randomly, so I've already explained that. I've already explained this common mistake here, which is the quadrat that they normally give you is 0.5 meters in length, so the aerial is going to be 0.25. Please don't presume it's 1 meter. Okay, the second practical. So once you've got happy with the idea of quadrats in that they're just a thing for looking at sampling the number of living things in an area that you've placed randomly, we can go on to transacts. So transacts, the whole point is to, this time, we're not looking at estimating a population. What we're doing with transacts is we're looking to see if something is affecting a population. All right, so the common example is this one right here, okay? So there is an area of a field and there's a tree, okay? So obviously this tree is going to be providing shade. So the light intensity is going to be different because the plants close to it aren't going to get as much light as the ones further away. So what you would do is you find an area that you want to investigate, you place a tape measure at 90 degrees to it. That's very important because actually we might be investigating the tree here, but we also might want to go out from the tree here. We might want to do 2. We might also want to go there, as long as you've got a nice straight line from it. So you've got a tape measure. And then what you do is you place quadrats at specific intervals. Okay, so you can see here I've placed it every 5 meters. All right, so like 5, 10, 15. So you just put your quadrat here, quadrat here, quadrat here, and then you just count. So let's say in this first one there was 1 daisy, in the second one there was 3 daisies, here there is 5, and then we did another one at 20 where there was also 5. So we can see, oh look, as we get further away from the tree along my transect line, the number of daisies is actually decreasing, and you can see that in the graph. Please don't say that because obviously here it doesn't. So you would say between 5 to 15 meters, the number of daisies is increasing. 15 and 20 meters, it's the same. What you need to figure out again is the factor. So why might this be happening? So you have a suspicion that it might be to do with the light because the tree is in the shade. So what you would also need to do, not only would you need to measure the number of daisies here, is you might need something called a light detector or a light meter, and the light meter just checks how much light is there in something called lux. All right, so you would measure the light intensity here. So you can see in the table here, I've done that. So the light intensity here was 2 lux, here it was 5, here it was 10, and here it was 10. So they could plot both of those on a graph, 2 different lines, and you can say very clearly that, look, as the light intensity increases, the number of daisies also increases. So the light intensity is a limiting factor. So here is the method. Place the tape measured perpendicular to the area being investigated. Place a quadrat at even intervals along that tape measure. You tell the examiner what you want it to be. It doesn't matter as long as they're even intervals. Count the number of organisms within each quadrat. Measure the light intensity using a light meter at the position of the quadrat. That is it. That is transacts. It's dead straightforward. They might link this to ask you reasons why. So for instance, if in this investigation, we can see that as we move further away from the tree, the number of daisies has increased. And they might say, give an abiotic reason and a biotic reason why that has happened. So remember, biotic are living things, abiotic are non-living things. So the light intensity is, you might say, oh, well, the tree causes shade, so the light intensity is increasing. That is a abiotic factor. So light intensity is an abiotic factor. But there might be other reasons. Maybe lots of animals go for shade under the tree. So actually the ground under the tree is all stamped flat by animals. So that's the reason why there's not many daisies there. So a biotic reason is lots of animals go under the tree for shade, therefore daisies in that area get stamped on. So there are lots of reasons that might be the case. Okay. I've just put a sample explanation here because you might be asked to explain a reason. So as the distance from the tree increases, the number of daisy plants increases. This is because the ground close to the tree is in shadow. As the distance from the tree increases, the light intensity increases. Light intensity is an abiotic factor for plant growth. Plants require light for the process of photosynthesis. So that's our explanation of it. Here is another one, Transect, and this was on a higher paper. It's a bit of a tricky question, this one. It's done Transect, so basically they've got the C and they've started their tape measure here. I'll use a different colour. They've started their tape measure here and they've placed their tape line, and what they've done is they've used quadrats all the way along and they've looked at these different populations as they've moved along. So they've done quite a lot of different tests and they've just recorded, so there's a little key down here, the deeper this line is, the more organisms there were in that area. And the wider the line is, the more places the organism was found. Okay, so we can see here that we're looking at it. The first question says, which organism was the most abundant? Abundant means more of them. So you can see, oh well, we're looking at the more organisms is the height. So the one that's got the biggest height consistently is this. So the toothed rock is the most abundant organism. The next question is, which area had the greatest biodiversity? Biodiversity means the number of living species within different species, within an area. So we can see, you just need to look through. So let me just get rid of my note on here. So here's an area where we have got 3 living things living there. We've also got 3 there, 3 there. Oh, but this area we have got 1, 2, 3, 4 different things. But this area here, we've got 1, 2, 3, 4, 5, 6. We have got 6 living things. So this area has the most biodiversity. Where is it? It's at the edge of the shallow rock pool. So the greatest biodiversity is at, and you need to say the edge because it's not in the middle of it, it's at the edge of the shallow rock pool. Suggest how biotic and abiotic factors could affect biodiversity. So, abiotic factor, you might say, oh well, at the edge of the rock pool, there's less water, so, and on here and here, there's going to be more light intensity, so that might affect biodiversity. Obviously, underneath the water, there's very little light intensity, so that's an abiotic factor. And biotic factors, you might say that maybe lots of, there are lots of living things which come to drink out of the rock pool, and therefore all of these things here get eaten. So the ones that live on the sand get eaten by predators, whereas the ones that are living in the rock pool can't get eaten by those predators. So that would be a biotic factor. Okay, so try and link the idea of abiotic and biotic. That's a common thing that they will do with sampling. All right, let's look at some common questions and then we'll have our break before we go into the paper 2 core content. So here's a common question. It's just calculating estimating the number of dandelion plants. It's worth 4 marks. It's a calculation. There's always worth plenty of marks. So we have got a 0.5 by 0.5 quadrat. So you should be going, okay, 0.5 times 0.5, which means we have got a 0.25 metre squared quadrat. If you have got that on your paper somewhere, you will get a mark. The mean number of dandelions in the quadrat is 2.8, so we don't need to calculate a mean or anything, that's given to us. So all we need to do is we need to use that formula. So if we want to estimate it, we need it's the area of the field or the sample divided by the area of the quadrat multiplied by the mean number of organisms in one quadrat. Okay, so that is the formula that we're going to use. So the area of the sample This is asking about field A, so we can see that field A is the field A is 120 metres times by 75.5 metres. Okay, that's going to give us our area. So let me just pull up a calculator and do that. That's not pulling up a calculator. This is pulling up a calculator. There we go. Okay, so we have got 120.0. If they're giving you the 0.0, they'll want you to use it. It's because they're telling you how many significant figures they're using or how many decimal places they're using. So your answer should have the same. Times by 75.5. So our area is 9,060 metres squared. Again, if you've got that somewhere on your paper, you'll get a mark. So what we need to do is do the area of the sample, so 9060 divided by the area of the quadrat, so 0.25. So 9060 divided by 0.25, which gives us 41181.8 quadrats. And we're going to multiply that by the mean. Now, this is the point where I would just be checking my answer, because that's a very funny number. Normally in exams, it all comes up, so I will just use that as an opportunity just to check it again. 120.0 times 75.5. Yeah, that's definitely what we got. And divide that by 0.25. Yeah, so that's what they're wanting. That says 0.22. Which one says 0.22, Chris? On the calculator. It does. You're absolutely right. Why have I written that? 0.25. Right, that's much better. I did that twice. That's weird to do the same error twice. Thank you for spotting that. That's why, if you get a result that doesn't look right, all right, the exams don't design things that way. Okay, so check it a couple of times. Thanks for spotting that, Chris. So we would get 36240. It's a nice round number. You can probably figure out, if you've got that you've probably done something correct. All right. So we'll have 36,240 quadrats would fit in that area. So all we need to do now is multiply with that by the number of organisms. So it's 36240 times by the 2.8, because there was 2.8 in every quadrat. 2.8, which is going to give us 101,472. So there is and the estimated value is 101,472 dandelions in that field. Okay, now I'm just going to go into this because they didn't, but they could have. Let's say with this diagram, and the AQA have a habit of using the same diagrams in multiple years and asking different things about them. If I'd drawn this diagram, I know what I'd be asking. I'd be saying something like this. In field B, the number of organisms per metres cubed or something, per metre squared, sorry, was less. Give abiotic and biotic factors to why that might be the case. So obviously they've put a great big hedge around it. So you could say that there are lots of things living in the hedge around field B which might eat the dandelion plants. That would be a biotic factor. Yeah, I can go over the calculation one more time. So they might eat that. Also, question, let's say it is recorded, so you can watch it back, and I'll put a solve in, but I will do it again quickly. Yeah. You could also say that the fact that the hedge is there, it's going to create shade, so that is an abiotic factor. So that would, if I was writing that question, I would not miss up the opportunity to ask about how the hedge might affect the population in field B. So it might not, but if they want to use that again, they might ask about that. Yeah, calculation one more time. Okay, so what we need to do is the area of the sample. So the area of the sample size was, we're given the two lems here. So I've calculated it here. So it's 120 times 75.5, which gives us 9060 metres. So 9060. We divide that by the area of the quadrat. We've already figured that out, which is 0.25. 0.5 meters squared because it was 0.5 times 0.5. That will give us an answer. You don't need to write the answer down, you could just press equals, and we're going to times that by the number of organisms in one quadrat, which is times by 2.8. So that's how we got the answer. Where do I get 120? I got the 120, Chris, because the field is 120 meters in length here. Okay, no problem. Please ask questions, because if one person's thinking it, it's quite likely that other people are thinking it too. Right, so let's have a look at the other one. So here you might be asked how to estimate. So this time, rather than getting us to actually do the calculation, we're just specifically being said, describe a method that a student could use to investigate the effect of a fence, around B on the population of dandelions compared to field A. Mention any equipment used. Now, I'm not going to write that down. It's a 6 marker. We need, again, to come up with a logical step-by-step method which makes sense, doesn't contradict itself, and definitely mentions equipment, because they've said that specifically, and would lead to a valid outcome where we'd actually be able to figure out the number. So all you need to say is this. So they would measure the length of field B with a tape measure. They would measure the width of field B with a tape measure, and they would find the area of the field using length times width. They would get a quadrat, and they would find the area of the quadrat. You don't need to say the area of the quadrat. They would then do the area of the field divided, they would then place the quadrat randomly in field B and count the number of organisms within the quadrat. They would repeat that 10 times. They would calculate a mean number of organisms in one quadrat. Then what they would do is they would do the area of the field divided by the area of the quadrat to figure out how many quadrats would go in the field, and multiply that number by the mean number of organisms in one quadrat. That will give them an estimated population size of the organisms in field B. Okay, so, and that would be worth 6 marks. If you're struggling with that one, if that one was like, ooh, went too fast, okay, as I say, it's all there, okay? It's in this booklet, which I am going to share with you, so just have a read through that again later, or rewatch the video back if you want to. Okay, the other thing you might need to do is to interpret some results from Transect. I've already showed you one result from Transect, and that's a really tricky one. That was a really high mark question. This one's not as bad. We're just given a graph. This was a distance from the river in meters. So obviously, basically, there was a river. Here's the end of the river. They put a tape measure, and they have gone every meter. So 1, 2, 3, 4, so on. And they have counted the number of different plant species using a quadrat at each step. And they have done it multiple times because what they've got is a range here. That's why I've put it in. Right, and they've presented their results on a table. So the cross is the mean number of organisms at each distance, and then we've got the maximum and the minimum here. All right, so this is showing us the range of organisms in that area. So the question says, what was the mean number of plants? What was the mean number of plants specific found at 3 meters from the river? I've written a spelling error. Species. What was the mean number of plant species found at 3 meters from the river? So all you need to do is draw a line up to the mean one, draw a cross, and we can see that the answer is 4. Which mean result has the greatest uncertainty? Now, uncertainty is a word that pops up a lot. Uncertainty equals the range divided by 2. Okay, basically it's the bigger the range, the larger the uncertainty. All right, so if we come out, I'm just going to take this line off to make it more clear. We're looking for the one with the biggest error bars. Okay, so that would have the biggest level of uncertainty. So we can see that this one, and this one have all got the same length error bars. They're all 4. So actually there isn't one result, it's 3, 4 and 6 have all got the same level of uncertainty. So it's just the range divided by 2. So 3, 4, and 6. A better question would probably be which result had the lowest uncertainty. So you can see that this one here has got a very small error range. So therefore, this is the result that is probably going to be the most accurate. Okay. What is the relationship shown in the graph? So in order to do that one, all right, it's just really common that we need to do is that we're going to just draw a line of best fit through the means, and we can see that as the distance from the river increases, the number of plant species increases too. It isn't a line going through the origin, so we can't say it's directly proportional. Suggest how biotic and abiotic factors could have caused this relationship. Hopefully you're spotting a pattern here. This is what we're going to ask you all the time. So again, same thing. So as the distance from the river is increasing, there is more plant species. So you would just say a biotic factor might be animals are going to the river in order to drink, therefore standing on the different plants. Therefore, that is killing some of the plants. And a abiotic factor could be water levels. So near the water, the ground is very wet, and the plants don't grow as well in wet conditions. Further away from the river, there's less wet, so the plants grow better. There you go. So you've got an abiotic factor too. And very finally, we will look at this one. This is the one where you have to figure out the area by doing that. You might be asked to explain the results of a sample. So there is an uneven distribution in the number of daisy plants on the school field. Explain how biotic and abiotic factors could cause this. Now, this one you can talk about loads of things. In fact, the picture is here just to give you some clues. Okay, so it's worth 6 marks. So you probably want to be mentioning a couple of biotic factors, and then it's about explaining them. So you don't need to mention 6 factors. You just need to mention a couple of factors and explain them. So you could say here, look, there's going to be shade. So you could say that some part of the school, the field, is in shade from the school buildings. The shade would reduce the light intensity. Therefore, the plants in that area would have less light to photosynthesize, so there'd be less daisies in that area. That would get you 2 to 3 marks because you've said what the factor is and you've explained it. You could say that on the sports pitches, lots of people are going to be running around playing sport. So you could say that on the sports pitches, there will be a high footfall or lots of people running on it. Therefore, the daisies would get trampled. So then the number of daisies there would be less. Make sure you say that that would be a biotic factor. So therefore, we've given an explanation for a biotic and a non-biotic. We could also say things like the trees are caused shade. So again, that's a biotic factor. Sorry, an abiotic factor. And if you absolutely wanted to, you could say that maybe some areas of the field might not be level, therefore there might be lots more water on them that would affect it too. All right, but just make sure you make a statement and then explain it. That's all I need to do for that one. Okay, that is the end of the focus on the required practicals. It took exactly an hour.